So real pleasure for me to introduce our keynote speaker, Peter Agre. Dr. Agre is a physician scientist and the 2003 Nobel laureate in chemistry. He is currently the Bloomberg Distinguished Professor and Director of Johns Hopkins Malaria Research Institute at the Bloomberg School of Public Health. Peter grew up outside of Minneapolis, Minnesota. Inspired by his father, a chemistry professor, the young Agre anticipated a career in science. But as he acknowledges in his very amusing and enjoyable Nobel biography, and if you have not read it, I would suggest that you Google that after his presentation. I've read it a few times, and I always learn something new when I go through it. What he notes is, and acknowledges very candidly, is that his academic performance was inconsistent. In fact, very candidly, he notes that in high school, he gravitated in a bohemian direction, discovering that by far the most interesting aspect of life was girls, followed closely by cross-country skiing, long-distance bicycling, and camping, which he did across Russia. By contrast, school was not the least bit interesting, quote unquote. This bohemian approach culminated in withdrawal from high school and after receiving a D in, of all things, chemistry. So for everybody out there, if you got a D in chemistry, you can go on to get a Nobel Prize in chemistry, so don't give up hope. So at this point, taking a short break from high school, he began to work as a truck driver for a factory manufacturing military equipment, and then went back and finished high school alongside his job in night school. As he writes, quote, "This experience in the real world as a truck driver was far less appealing than I had anticipated. And this, I suspect, served as a real inflection point for him to double down and pursue his studies." And he did exactly that. Dr. Agre studied chemistry at Augsburg College and pursued his medical doctorate at Johns Hopkins University. He completed his residency at University Hospitals, Case Western Reserve University in Cleveland, and oncology fellowship at UNC Chapel Hill. He joined the Johns Hopkins School of Medicine faculty in 1948 and rose to the rank of Professor of Biological Chemistry and Medicine. Peter is best known for the discovery of aquaporin water channels, the family of membrane proteins that regulate the movement of water in and out of cells. Aquaporins are found in all living organisms, from microbes to plants to mammals. Just to underscore the importance of the work, remember that about 60 to 70% of the human body consists of salt water, that the body's cells must contain specific channels for transporting water in and out was suspected as early as the middle of the 19th century. But it was not till 1988 that Agre and colleagues succeeded in isolating a membrane protein and thereby offering decisive proof of the existence of the long-sought water channel. In humans, aquaporins control water balance in the kidney, in the cerebrospinal fluid of the brain, sweat from skin, and secretion of tears and humor, aqueous humor from the eye. And dysfunction of these channels has been linked to a wide spectrum of diseases. I'll conclude by simply noting that Dr. Agre's contributions extend far beyond the laboratory. He has and continues to serve as a powerful advocate for science, and he gives incredibly generously of his time to support these activities. As an example, as president of the American Association for the Advancement of Science, AAAS, he traveled to Cuba, North Korea, Myanmar, as a member of a scientific delegations tasked with finding common scientific ground with investigators in these countries. And more recently, and something that was very, very meaningful to me personally, Just before the pandemic, he was kind enough to offer his support for a conference that I had organized at the National Academies in Washington, D.C. That conference was focused on seeking novel solutions to augment the number of physician scientists in our nation. It's something Peter's very passionate about. And so with those few words, I'd like to welcome Dr. Peter Agre to deliver this year's keynote address entitled From Lake Wobegon to Stockholm through Adelbert Road. Peter.
Good morning. Is my voice coming through?
I can hear you loud and clear, Peter.
Excellent. Thank you, Mukesh, for the introduction. I sometimes like to forget some of the wayward part of my youth, but it's all basically fact. I'm going to concentrate my talk today on not the technical side of the science that we pursued over the years, but the people who worked on the projects and the amazing people I've met during my career, starting with these two people, Mukesh, you're a rising star in cardiology and medicine, and we wish you the best as dean at the Warren Alpert School of Medicine at Brown University. And Michael, I oftentimes think of you as the Bruce Springsteen of medicine. Absolutely no pretense, an intense message which comes year after year. So heartening. I'd like to dedicate this talk to Charles Carpenter. Chuck Carpenter was the chairman of medicine when I was a resident at Case, and I'll talk about Chuck and his incredible life, as well as the other people. So I attended medical school at Johns Hopkins, and I was particularly eager to attend Hopkins because they had a strong reputation for being an institution with global health programs around the world. Medicine for the developing world was something rather new, and it was Chuck Carpenter, Brad Sachs, shown in this photograph, that established the Diarrheal Research Center in Calcutta and subsequently moved to Dhaka, Bangladesh. They were only in their early 30s, and they moved their families, small children, to Calcutta for 2 years to pursue cholera, and were early in the development of the oral rehydration therapy. And this is the sort of work that I thought, growing up in Minnesota in those cold winters, would be a very exciting and wonderful career. So I departed Hopkins in 1970, made my way east to Johns Hopkins by going west, traveling through East Asia, Southeast Asia, India, Middle East, seeing sites of squalor and splendor. There was much to be learned. And of course, stopping at some of the research stations that Johns Hopkins had established along the way, I was firmly entranced. When I got to Hopkins, as a medical student, had the great fortune of having a roommate, a friend I made in our class of medical school, Van Bennett, who steered me towards the laboratory. Cholera toxin had just been isolated in 1970, and it was a very big advance. This is work of Richard Finkelstein at the University of Texas. And it provided a clue as to the mechanisms for other infectious diarrheals, like the traveler's diarrhea, which was believed to be an E. coli problem, but no one had isolated the toxin. So I worked with Pedro Quattrocastis, a young faculty member at Johns Hopkins, himself an MD, WashU graduate, Hopkins residency, trained at NIH with Chris Anfinson, and used affinity chromatography for the first time in biological systems. And they used it to isolate the estrogen receptor, work with Gianfredo Pucca, a visiting professor from Italy, shown here. The rest of the laboratory was a lot less photogenic. This was our group in 1974, including scientists from around the world. You notice there are no women. There's been a big change in science. The appearance of women in science is changed things quite a bit. It's really redoubled the workforce and has led to great things. So as a rather immature student, I wasn't quite sure how this was going to unfold. I remember attending a mixer at Goucher College, meeting an attractive young lady, and we danced, and she asked me what medical specialty I would be interested in. And something told me I should say neurosurgery or cardiology, but I'm from Minnesota. I told her the truth. I said, I'm interested in diarrhea. And that was quickly the end of that potential romance. But I soon thereafter met a technical assistant at Hopkins. She grew up on a farm, and that's Mary, my wife. And after graduation from medical school, I had the great fortune of joining the University Hospital Department of Medicine residency, led by none other than Chuck Carpenter. We referred to him as the tall man. He clearly towered over us. I was standing on the second row, in the second row, and we're on the second step. He was probably a 6'5" tall gentleman, and he put his imprint rapidly on the house staff training at university hospitals. He increased the presence of women. There were only, in this photograph of our house staff, there were, I think, 49 heads I counted, and there were only 4 women. But they increased the participation of African Americans. And of these people in the photographs, 11 were from Johns Hopkins. He was like a magnet. His generous spirit and high standards were ever so important to development. So here I am in the background, in the shadow of Chuck Carpenter. Chuck, in addition to strong devotion to the care of patients, was always insistent on the highest quality of clinical care without regard to patients' socioeconomic status, race, sexual preference, gender. It was something that all patients deserved, and he insisted that they would receive it. And he would often be in the wards late in the evening if there was a problem to assist or encourage people. It was really Chuck's encouragement that led me to pursue biomedical research in a clinical setting. So he was a Case Western and university hospitals for 13 years. He arrived in '73, and in 1986, there was a dispute with regard to new building, which was to provide laboratory space for clinical investigators that the dean had repurposed for private outpatient medicine. It caused Chuck to resign his position as chair of medicine. He did a short sabbatical at the NIH with a young scientist named Anthony Fauci, and took the chairmanship at Miriam Hospital at Brown University, and started the Center for AIDS Research at the Providence Boston program, which he worked for the next 30 years. He concentrated on HIV infections in women, which were at that time really underappreciated, and developed a healthcare program for incarcerated women in the Rhode Island prison system. He carried that spirit with him to the very end. This is a picture of Chuck and me at the Brown Medical School Research Day 2 years ago. He passed on about 9 months later, but a gentleman to the very end. There were other fascinating individuals I met at my days at University Hospital. Oscar Ratnoff, a giant in the blood coagulation field, discoverer of Hageman's factor, shown on the left is Dr. Ratnoff with John Hageman, a trained brakeman who was deficient in Hageman factor. Ratnoff preferred to call it by the name of the proband rather than the Roman numerals that he finds oftentimes people would mix up. And on the right is Dr. Ratnoff as we saw him, an older man. he would have rounds, which we, of course, very sarcastically refer to as rat rounds, but he would share scientific insight to a clinical problem and oftentimes a personal story. Fascinating man. Adel Mahmoud, very elegant Egyptian from Alexandria, infectious disease attending, went on to become the interim chair of medicine at university hospitals, and then became the president of the Merck Vaccine Institute, brought forth the human papillomavirus vaccine. All didn't turn out as expected. This is Tom Butler, another infectious disease attending. I met him when he was doing his fellowship at Hopkins, and he was in the faculty at Case Western. A very egregious miscarriage of justice occurred in 2003 at the time after the 9/11 attacks, and fears were high, and he was charged with lying to the FBI when some vials of plague bacillus disappeared from his laboratory. Of course, he was acquitted of that, but when the FBI comes after someone at a time of a national emergency, bad things can happen. Tom was sentenced to 2 years in prison for some simple mistakes on the forms that he had to fill out to ship the samples. And many of us stood up for Tom. Here he is on the 60 Minutes news magazine. But in the end, he took his punishment. And after release from prison, he rejoined the academic medical community. He's teaching at one of the medical schools in the Caribbean. I think this is a reminder to all of us that rules, even though they seem onerous, must be attended to. I keep in touch with Tom. He's a wonderful man and a great colleague. I had the fortune of my senior year as a senior resident to spend a couple of months in the laboratory of Jim Carter. I couldn't find a picture of Jim. He's an endocrine faculty, and his associate, Viswanath Chandramouli. Of course, we called Mouli by his preferred name, which is just Mouli. I really learned a lot from Mouli, and he had a delightful sense of humor. Very dark-skinned South Indian. In the summer, his skin tone was very black, and people would sometimes ask him his background, and he would always answer, "I am a Cleveland Indian." Great sense of humor. I talked about Mike a little bit. I didn't share the extracurricular activities that some of the House officers participated in, most notably a soccer team, which Mike organized. He was an athlete at Brandeis, and we had a soccer team known as the Old Timers. Our jerseys were purloined surgical scrub shirts, and we had a lot of fun playing soccer. Some of the regulars were Al Braverman, who went into gastroenterology, David Wright from neurology. Of course, Michael was there in the center of it all. Another colleague, Gordon Ginder, and I were classmates at Hopkins and residents. He's gone on to a distinguished career in hematology oncology and has served as the director of the Massey Comprehensive Cancer Center at Medical College of Virginia for more than 20 years. And the students, some of the students turn out to have spectacular academic careers doing emerging research with clinical medicine, true physician scholars. Mark Nepper, who's the director of the Laboratory of Kidney and Electrolyte Metabolism at the Heart, Lung, and Blood Institute. We've collaborated. It was Mark who coined the term aquaporin. Skip Brass, a specialist in platelets and authority on the role of platelets in stroke, has directed the University of Pennsylvania MSTP program for more than 25 years, probably the best in the country. And Helen Hobbs, these were all medical students on the wards at University Hospital when I met them. Helen has gone on to be a distinguished Howard Hughes medical investigator, worked out the genetics of hyperlipidemia, Breakthrough Prize winner, Harrington Prize winner, Asano Award lecturer, clearly a scientific star. And these are the people that I was so pleased to know early in their careers and still maintain contact. So following University Hospital and a fellowship at UNC Chapel Hill in Hemong, I turned to Johns Hopkins and joined the faculty and had a small lab. It was basically a two-person tiny laboratory, and it was in the 10th floor of the Blaylock building, which was the same floor as the Department of Medicine headquarters, the chairman's office, and there were no other laboratories next to us. But I was able to entice some wonderful people to join us. Brad Denker, who's now the head of nephrology, clinical nephrology at Beth Israel Hospital at Harvard Medical School, and Barbara Smith, my technical assistant, together identified a protein in red cell membranes that no one had seen before. The Kumasi stained polyacrylamide gel on the left shows in lane 1 whole red cell membranes. And when the same slab is stained with silver reagent, you can see many proteins that don't show up by Kumasi, including in lane 4, a protein that fairly stains with Kumasi that turned out to be a protein we could isolate very simply, a simple detergent extraction. And it was present in a large number of copies per red cell. We were very excited, but we didn't have a clue what the protein did. We studied it biochemically and biophysically, but we didn't have a functional insight. So I did a sabbatical, learned the DNA technology, cloned it out, and saw that the genetics database, this is now 30 years ago, which for me seems like pretty recently. For the young people, it was ancient history. It had some relatives in the database from diverse sources, but none were functionally understood. Protein from lens, protein from Drosophila brain, a factor that allows bacteria to metabolize glycerol, and some gene fragments from plants. We were really stuck, didn't know how to pursue the project. We figured it was a bilayer spanning domain and had an interesting topology with an internal repeat, which is obversely oriented, the amino terminal half to the carboxy terminal half. Well, the solution to the mystery turned out to be a lucky result of a family camping trip. My wife and I have 4 wonderful children. They're grown now in their 30s and early 40s. But when they were growing up, we would camp in a national park every summer. And after doing this several summers, Mary asked the kids which national park they'd like to go to next year, and they said Disney World. So it was a trip to Disney World, but we stopped in Chapel Hill, North Carolina to visit my colleagues, and I discussed with this individual, John Parker, eminent hematologist, investigator, the nature of this 28 kilodalton protein we'd isolated, which we really were stuck. And John listened and he said, it's present in red cells, renal tubules, homologs in plants. He said, well, you know, these are the most water-soluble membranes. Maybe this is the long-sought water channel. So this was really John's idea. And he explained how water is homeostatically at equilibrium in most of our tissues, and things proceed in very orderly. But when water is in disequilibrium, as in brain edema or glaucoma, renal failure, it could be a very dangerous substance. And the movement across cell membranes was thought to be governed by membrane channels, water channels, but these were hypothetical. No one could isolate them. And we set about establishing this in collaboration with Bill Gugino. Bill was a young man at the time. We were both had just turned 40, and Bill's lab was studying the cystic fibrosis membrane transregulator by expressing it in Xenophus laevis oocytes. These are frog eggs. So we did a simple experiment, injecting a control egg on the left with buffer alone and the test egg on the right with the cRNA expressing this new protein, and transfer them to hypotonic solutions. The test oocyte exploded. it was the first experiment that really showed that this was, in fact, the membrane water channel. And this produced much jubilation in the laboratory. Greg Preston, a postdoctoral fellow who did many of these initial studies, he was jubilant, and with good reason. I have to admit, I took this photograph of Greg 2 years after his report came out in Science, and he was still celebrating. I'm going to quickly step through the next slides because the important information has all been published, but we set about solving the structure in collaboration with these two scientists, Yoshinori Fujiyoshi from Kyoto University and Andreas Engel from the University of Basel in Switzerland. And Andreas, in retirement, has worked in the faculty of Case Western Reserve University in the biophysics group. And it was really their pioneering efforts that allowed us to solve the structure by cryo-electron microscopy. And the answer is that the protein is tetrameric with a single channel down through each subunit. If you look to the left, this is a channel subunit when viewed from above. There's a single pore in the center, about 3 angstroms in diameter. And on the right is a cross-section through the membrane. You can see a single channel. There's a little side to side, but some key residues were identified. And this explained how water crosses the membranes of some tissues with almost no resistance. So water is a fascinating molecule, H2O. You'd predict it to be a gas, and of course water is a gas in the atmosphere. But in bulk solution, the hydrogen bonding between water molecules causes it to actually form a fluid, which would be better explained as H2O to the nth power. So the aquaporin, as we've named these, has water in bulk solution in the extracellular vestibule and the intracellular vestibule. In the very center of the channel, there's a alignment of water, which is hydrogen bonded to the adjacent molecule, except at the very center of the channel where there's this internal repeat. We refer to this as the hourglass. And the water molecules at that point, as shown in the lower panels, will hydrogen bond to the two conserved asparagines, isolating a single water molecule from the column, preventing the movement of protons. This is important. It allows us to reabsorb water, but not reabsorb protons, H3O plus, the hydronium ion. Thus our kidneys can conserve water and excrete acid load. And this is work of David Kozino, a graduate student, now assistant professor at Dana-Farber Cancer Center at Harvard Medical School. Soren Nielsen, a young Dane who just completed his training. He was 29 years old when we started collaborating. He was a superb microscopist. And we worked together for 15 years, establishing the presence of the aquaporins in different tissues. His greatest interest was kidney, where the proximal nephron is known to be constitutively permeated by water. So in the diagram on the left, you can see the proximal nephron is highlighted in green. And on the right, the meta-analysis of multiple studies showing constitutive, and it's a logarithmic scale, constitutive water permeability. But the surprise was the collecting ducts. We found no staining with our very specialized antibodies, and there were multiple antibodies that we had to AQP1, but it never labeled the collecting ducts. So shown here on the lower left is a proximal tubule from outer medulla of a rat kidney. The primary urine flows through this space marked by a P. The membranes at the apical surface are jam-packed with aquaporin-1 protein, as are the lateral and basal membranes. But the collecting duct, marked C, fails to stain, indicating there must be another member of the family at that location. And I'll tell you in a moment that's correct. So this explains how water can cross an epithelial barrier following the small standing osmotic gradients that salt and sugar transporters create. Now, as a physician scientist, I, of course, was very interested in the human pathology that we expected to find with aquaporin 1. And I had a masterful postdoctoral fellow in the laboratory, Landon King, who is the chief resident on the Osler surface at Johns Hopkins and doing a pulmonary fellowship. He wanted to work on aquaporins in lung, and shown with permission as a French school teacher in the center who was identified as being colton null. We localized the structural aquaporin 1 gene to the short arm of chromosome 7, which was the precise location of the colton blood group antigens. And on the far left is Melanie Bonhever, a French-speaking postdoc. So with great care, we did clinical phenotypings on this and 2 other individuals that we identified as colton null from the International Blood Group Referencing System in Bristol, England. And the blood group, the transfusion medicine blood group experts are very altruistic. They share information, and they send us red cells, DNA, and urine from their patient. And we found that the individuals were unable to concentrate their urine. Normally, individuals, when they retire in the evening, will sleep 7 hours or so and awaken in the morning fluid deprived. And of course, to compensate for this, our kidneys concentrate the urine to about 1,000 milliosmolar. The colton null individuals, which we established were aquaporin 1 null, they had knockout mutations, frame shifts, deletions, and each family being a different, but it was a recessive trait. And the null individuals can concentrate only up to about 420 milliosms. The protein, aquaporin 1, is also very abundant in capillaries. And in a study with Robert Brown and Landon, they established that there was a decrease in the fluid uptake in the pulmonary vasculature in the lung, which was found in the aquaporin null individuals. Now, the individuals that I'm talking about with aquaporin 1 deficiency are clinically normal unless they're stressed. There was a fascinating paper in the New England Journal the week before last by Olivier de Voist, a colleague from Belgium, who in a large European study established that promoter variants in aquaporin 1 are found in individuals who are resistant to peritoneal dialysis. So I think the clinical phenotype is still emerging. So we were very excited as the interest in the aquaporin 1 rose, and investigators in multiple laboratories around the world by homology cloning identified 13 human aquaporin variants. And I'll tell you briefly a little bit about each, but mostly about the people who did the studies. The aquaporin 2, as I mentioned, is a regulated water channel, and it was Mark Knepper. I showed his photograph. He was a medical student when I met him at Case Western in the University Hospital wards. Mark Knepper led the group that worked out the biology behind vasopressin regulation of aquaporin 2 in renal collecting ducts. It's basically, if you can look at the cartoon on the right, it's an intracellular vesicular protein which traffics to the apical surface in response to vasopressin through an adenylate cyclase protein kinase A mechanism. Masato Yusui from Keio University in Tokyo worked in our laboratory on the lens homolog. We call it aquaporin 0 because the protein was identified before we got involved, but the function was not known. And mutations in the aquaporin 0 gene lead to congenital cataracts in small children. A study in collaboration with Shomi Bhattacharya and his team from London's Moorfield Eye Hospital. Aquaporin 4 in brain is a protein which I think is going to have profound significance, particularly in neurology and neurosurgery. In collaboration with Ole Petter Ottersen, who at the time of our studies was a neuroscientist at the University of Oslo. He's now the rector of the Karolinska Institute in Stockholm. But the studies we undertook with Søren Nielsen and Ole Petter established that the aquaporin 4 water channel in brain resided at a very interesting and very specific location. Capillaries in brain are surrounded by a basement membrane and astroglial end feet. On the diagram I borrowed from Kandel's neuroscience book, it shows the orientation. And on the right are micrographs, immunogold electron micrographs that Ole Petter's team had prepared, and they found that the gold staining, the protein is very abundant in the pericapillary membrane of the astroglial end foot, but it was relatively absent in the other membranes of the cell, so it's polarized in its location, which is precisely what you'd expect if it was important in the manifestations of brain edema. Of course, brain edema is tragic and it's very common. Closed head injuries, athletic injuries, stroke, brain tumor. It is responsible for tremendous human hardship. But the neurosurgeons are quick to point out that they have no good treatment for cerebral edema. So Mahmoud Amiri, who was a medical student in Ole Petter's lab, led a series of studies. I just have to mention a little bit about Mahmoud. His path to neuroscience was very indirect. His family was Iranian. They fled Iran at the time of the Islamic Revolution. And so he and his brother grew up in a refugee camp in Pakistan that fortunately was adopted by the Norwegian Social Services. And as a student in Oslo, he was magnificent. He led a series of studies in collaboration with our team in Baltimore using a special knockout mouse created by Stan Frainer and Marv Adams at the University of Washington. The targeted gene is the syntrophin molecule, which brackets aquaporin 4 at the blood-brain barrier. And he did a very simple study. He compared normal mice to the aquaporin 4 mutant mice and administered a defining brain injury, a 30-minute ligature on the temporal artery of mouse, reperfusion for 24 hours, sacrifice. And what you should see is that the normal mice have extensive edema and infarct. The rose color is vital staining. The mutant mice are relatively spared from this. And this has been reproduced in variations by laboratories around the world. If we could develop an inhibitor, this could be a potential clinical management for brain edema. And I have participated with a group that Walter Boron, who's in your physiology department at Case Western. Walter and his colleagues started a company called Aeromics and had discovered an inhibitor of aquaporin 4. It was in development, but further analysis has showed that the actions on aquaporin 4 are almost certainly indirect. And there are other pathways which are targeted. So the lead compound is now being reevaluated, but the approach is very clear and the need is enormous. Another member of the family, aquaporin 5, coded by Surbi Raina, when she was in our laboratory. Surbi is now a professor of biology at Syracuse University in New York. And this is present at the last membrane water crosses during the generation of salt, saliva, tears, and sweat. In a mouse study with Lenny Nasum, a young Danish investigator, we established that, in fact, the generation of sweat depends on aquaporin 5. On the left is a normal mouse paw coated with starch and iodine, and animals injected with pilocarpine. And the amylase in sweat degrades the starch, which reacts with iodine. So the blue dots represent functional sweat glands. On the right, you can see that the sweat glands are hypofunctional. They're present, but they don't function effectively. And of course, mice live in the cool shadows. The humans labor in the hot sun. The ability to sweat is essential. Briefly, there are another branch of the family, which we've referred to as the aquaglyceroporins, and the structural studies reveal that the pore of the aquaglyceroporins is a little larger. I won't get into that, but this is a collaborative study with our structural biologists and Robert Stroud and Bing Yap, two investigators in the University of California, San Francisco, and at UC Berkeley. The aquaglyceroporin 3 is present in skin. This is a study that Johan Ogren prepared while he was a sabbatical worker in our laboratory, and the basal levels are intensely labeled. It didn't take long for the beauty industry to get interested in this. Some executives from the Christian Dior company came to visit me and invited me to give a lecture in Paris. They had a product which they would like you to buy called Hydraction Skin Cream. And their chemists had identified some small naturally occurring molecules which appear to increase the expression of aquaglyceroporin 3 in skin. Of course, the beauty industry is not held to the rigor of the pharmaceutical development, so you'll have to take their word for it. The implication that if you buy, so it costs about 50 euros for a 50-gram jar, you'll look like the model in the photograph, is hardly proof. But the advertisers are very bold, profound hydration, spectacular results. And what's this at the bottom? A Nobel Prize in chemistry. I showed this to my mother. She was at the time in her late 80s in a nursing home in Minnesota. And she saw this and smiled and said, "Peter, I think you're finally doing something useful." Our mothers know us very well. I hope we're doing something useful. I'm not sure that the Hydraction Skin Cream is the most important. Two others I'll briefly mention: aquaglyceroporin 7 in fat, aquaglyceroporin 9 in liver. The mechanism by which fat, energy stored as fat, is recalled. There's hydrolysis of the lipids in fat. The fatty acid molecules are transported by specific transporters, and the glycerol is released through aquaglyceroporin 7. It's taken up by the liver through aquaglyceroporin 9, which is upregulated during fasting. Human mutants have been found in aquaglyceroporin 7 in Japan, and they appear to have no phenotype. But when exercised to exhaustion, they fail to release glycerol from fat. So the process that marathon runners are so familiar with, refer to as bonking, is the conversion from glycolysis of glycogen to burning glycerol from fat. And so we know these individuals, despite their normal phenotype, will not win the Tokyo Marathon because of this deficit. And of course, during human evolution, the ability to store and recall energy is a survival factor. This is work of Jen Carberry, a grad student, postdoc who's now an assistant professor at Duke Medical Center in the cell biology program. Barry Rosen and his team at Wayne State were studying arsenite transport in yeast and discovered that the aquaglyceroporins are very efficient in transporting arsenite. At neutral pH, arsenic is uncharged. It must resemble glycerol. And in fact, arsenic is a huge concern in terms of drinking water. Fortunately, in the United States, our drinking water is, for the most part, very clean. But arsenic has not been a problem. In Bangladesh and Eastern India, we hear the sirens of Baltimore. It'll pass. In Eastern India and Bangladesh, where cholera is prevalent in the surface water, the villagers will tap groundwater, which is contaminated with arsenic. It's a huge public health problem with the emergence of tens of thousands of cases of hepatocellular carcinoma over the years. In closing, I just mentioned aquaglyceroporin 3 is also present in red cells, where it plays a role in the growth of malaria parasites, work that Dominique Promeneur led. So for the myriazoa in the ring stage to grow and divide, the glycerol transport requires the aquaglyceroporins, and the targeted parasites will still grow and divide, but with lesser virulence. Here you can see, just left of center, single ring stage malaria parasite in a red cell, and on the right, Schizont, where it has devoured the hemoglobin and grown and divided. And of course, when the Schizont ruptures, 32 new cells will be infected. Mosquitoes also have aquaporins in the Malpighian tubules. I won't go into that, but we'll just mention that one of the wonderful opportunities which I have had and have enjoyed so much as director of the Malaria Institute is to work at our field stations in southern Zambia. The Johns Hopkins Malaria Research Institute was made possible by a generous gift from Michael Bloomberg and the Bloomberg Philanthropies. And here's our team in southern Zambia, young Zambians and Zimbabweans who were working their hearts out on the malaria problem. And by simply working in the area and diagnosing, aggressively treating, malaria has come under control in the district around our main field site. There's been a 95% reduction in the burden. And this is not totally understood, but it clearly reflects the approach of modern medicine in settings where life is very challenging. We know it'll happen, of course, if we end the efforts, malaria will come back. Finally, an illustration of aquaporins in plants. On the right is a Arabidopsis. This is a photograph of Ralph Kallenhoff from Würzburg, Germany sent me. And the Arabidopsis on the left was an engineered plant which had disrupted the rootlet aquaporins. There are 35 different paralogs in Arabidopsis, but one in particular reduces the rootlet water uptake by about 90%. And the plant is keenly aware of this. It compensates by sending out a greatly arborealized rootlet system to provide the water necessary for maintain the succulent foliage. So of course, in the droughts in the plains of Eastern Africa, the water holes dry up, the animals wander off in search of water to another water hole, but the plants are left behind, and the plants compete for water. So Mukesh has asked me to talk about the Nobel, and I have to say there was a lot of excitement. early in the morning, the phone rang, a voice with a Swedish accent said, "This is an important telephone call from Stockholm for Professor Peter Agre. Are you Professor Agre?" I said, "I sure am." And he put me through and they informed me that I would share the Nobel Prize with Roderick McKinnon. And then they said, "In 10 minutes, we're going to have a press conference. You should be ready for your day." Congratulated me. And I ran off to the shower. And Mary, my wife, called my mother back in Minnesota. She was living alone. My father, a chemistry professor, had died 8 years earlier. And she shared the news. My mother, in her usual Nordic understatement, said, "Well, tell Peter not to let this go to his head. We're still waiting for him to do something important." It was a lot of fun. We enjoyed the reverie in Stockholm. The celebrations started almost immediately in Baltimore. The Cut Rate liquor store near my house was congratulating me. And I have to point out that the implication that I was their best customer is gross exaggeration. The laureates will spend a week in Stockholm. This year, of course, they will not because of COVID, giving a formal lecture on the important science behind the prize. And then in the most formal settings, at the Symphony Hall, the laureates are called up, and here I am with Rod McKinnon from Rockefeller University. And one at a time, the laureates step up and shake hands with King Carl Gustaf. He's muttered something in Swedish, and no one has ever figured out what he's saying. But then the laureate's job is to bow to the king, bow to the laureates, bow to the audience, and sit down. It was a bit of an out-of-body experience. My wife, Mary, holding the medal, I had to touch it to make sure I knew it was real. And here we were on the stage, my 4 children, Mary, and the medal. And I just like to point out that while the Nobels are awarded to individuals, it really should be viewed as an award to the field. The field of transport biology has come so far, and in the aquaporin field alone, there are multiple laboratories in several different countries, and really it's the combined effort that makes it important. At the end of the banquet, following the ceremony, there's a sit-down dinner for 1,200 people in the city hall, and this service is often provided by opera singers who are singing beautiful music. It's hard to behold such an occasion, but each prize is given the opportunity to make a short speech. And it's the older, if there are multiple winners of one of the prizes, it's the older laureate who will be asked to give a speech. And so I was asked to give a speech, and I decided to refocus not on the work that we had done, but the value of scientific collaborations and how science has grown so broad that individuals can never have all the technology. And then I dedicated to the men and women who teach science to the children in our schools, which got a very good reception. And we danced the night away. So Mukesh has also asked me to say a few words of advice to the young people. And my advice is, science is a difficult career, but it's a beautiful career. And looking back now at the end of my career, I'm pleased with all of the discoveries our team has made, but I'm equally pleased by the friendships that this has brought, the people that have come to our laboratory from other laboratories, collaborated, and even our competitors. We really have done something special that reflects all of us. Science is very much a social event. So the words of advice can be found laid out very clearly for you in a small volume that's still in print, Advice for a Young Investigator, translation of the writings of Santiago Ramon y Cajal, who won the Nobel Prize in 1906 in physiology or medicine, for his studies of the nervous system. Here are his pen and ink drawings of Purkinje cells. And Cajal said, there are no small problems. Small problems are large problems that are not understood. And I think in my experience, that's clearly the case. This simple issue of water permeating cell membranes has turned out to have a complex and fascinating series of biological systems that are involved. And the final word is Wei Ji, Mandarin for crisis. This is something one of my Chinese postdocs like to share. Wei Ji was actually two characters. Wei, a time of danger, Ji, a time of opportunity. And I don't think it could be said better than that. Certainly in our present day COVID pandemic, the dangers are real. But the opportunities were pursued very actively by multiple investigators, and the triumph of science brought forth vaccines which are effective and are being distributed. And I think the public is reminded once again that investment in science will make better lives for ourselves, our children, and our grandchildren. And with that, I'd like to thank Mukesh for the opportunity of presenting the lecture. I regret that it has to be a Zoom lecture or internet, and I look forward to the next occasion when I can stop by university hospitals and say hello. Thank you so much.
Thanks very much, Peter. That was really inspiring. And thanks so much for sharing the personal aspect of science in your journey, as well as the science itself. And I really want to thank you. You know, that comment that you made at the end about Nobels, and that they really should be given to the field, because none of us work in isolation. And it's a team effort, and it's a team of scientists from across the land and globe that bring fields up and recognition to it. So thank you for doing it so beautifully today. We do have a few questions, and I want to start with one from an alumnus of St. Olaf College. How brilliant is that, right?
Right.
And this individual asks, what advice would you give regarding balancing basic science research, teaching, and clinical work? Thank you so much for your inspiring talk from a St. Olaf College alumnus and UH psychiatry resident. So the question is balancing research, the tripartite mission, essentially.
Well, it's a big challenge, and very few people succeed at all three. They're equally important. I mean, Chuck Carpenter, Mukesh, Mike Letterman, there are people that are outstanding physicians, outstanding scientists, and outstanding teachers. But the day is short, and I tried to be fair in my career, particularly the balance with having a family. And I think there has to be some limits. If we worked all the time, we could do more experiments, but you can't do all of the experiments. You have to be selective. And so I think it's constantly important to be honest with yourself and question the validity of the projects you're working on. Are we really doing as much as we can to disprove our own hypothesis? I mean, this is Sherlock Holmes. When you exclude all of the suspects, the one remaining is the criminal. And sometimes discoveries are that way.
Yeah. So, and maybe there's several other questions, Peter, but I'm going to sort of consolidate them into one. They're all related essentially to the physician-scientist career path, and that physician scientists have played a critical role in discoveries, but they account for less than 2%. And your thoughts on how we can increase the workforce or encourage individuals to go into science?
Well, I think the emergence of women in medicine and in science has really been a major change during my career. I showed you our laboratory in Pedro's lab in 1974, no women. Now our graduate programs have at least 50% women. In our clinical programs, the interest is still larger. I think setting examples is important. I think when we are frustrated with our challenges of raising grant money, we sometimes discourage the younger scientists, and that's most unfortunate. I think physician scientists are essential to bring discoveries to the clinic. I think the background as a physician is very precious, and certainly it helped me remarkably in my career as we looked at different aquaporin paralogs. We had a pretty good idea what they would do. And I think that the country and the world is really dependent on physician scientists. And I think the rewards are not financial, but in terms of the well-being of society, they're really essential.
Well, thank you, and thank you. Your comments are spot on, and they also remind me to thank you on behalf of the MSTP students and the PSTP residents and fellows that you met yesterday. They were just absolutely blown away by the opportunity to meet with you and to learn from you. So thank you for serving as an inspiration for the young next generation of physician scientists in our community.
Thanks, Mukesh.
And I want to, Peter, we're going to have a few things to send you, which we will, of course, mail to you. But there's one that I do have here that I want to share with you and the audience. And I'm going to try not to mess this up and be all thumbs. And I'm in a position where I actually do not know if I am holding this high enough for everyone to see, because I can't see anything in this world, but you can see it. So this is our effort, all right, our effort to represent aquaporin 1 crystal structure in a crystal cube.
Wow.
And it reads, it of course has, distinguished scientist lecture, today's date, your name, for delivering the keynote address. And just in case you forgot or thought we were off a little bit, we called it the aquaporin 1 protein. So we'll send this along with a few other things to you and to Trish, who's been wonderful. And thank you so much for delivering today's keynote address.
Well, I'm delighted, I'm humbled. Thank you from the bottom of my heart.